There we go. Hi, Grady. You can say hi. Everyone say hi.
Grady, they put speed bumps in.
Yeah, they put speed bumps in.
We're going to start a revolution.
That's right. We're all whining about the new speed bumps.
Moving to Texas. We're moving to Texas.
Just saying, I saved Natalie's life yesterday because I slowed down over them and she didn't see them.
I didn't see them.
How did you not see them?
I can't see over her. I can't see
Well, I did not see them. I would have just kept going the same speed, and I would have not even been alive today.
I know. Wow. All right, guys. I'm going to plug along. I have good news and bad news. Actually, it's okay. I just need to plug along because next Tuesday there's a good chance I will not be here. I hate missing school, but my little kid is going to the zoo, and they are in desperate need of chaperones. And I usually don't like to do that, but being the good mommy that I am,
Sometimes the zoo is more important.
Actually, I think I'd rather hang out with your guys' age groups than about like 56-year-olds. So I. Hey, but don't fret, because maybe what I might do, if we can't get through this material in time, I may do this thing and have you guys watch it while I'm gone. We'll see.
You're going to teach us at the zoo?
I may be at the zoo, and you may be watching a recording of Mrs. Hatcher during this time. So let's see how fast we can get, or far we can get through the material. I'd rather do it live, but you know, we always have that option. I realize next week is weird too because you don't have school on Friday. And so I want to get a test in on that Thursday. I really want to get a test in on Thursday. That's kind of why I'm aiming. And then that whole Tuesday thing kind of screwed up. All right, so let's plug along. Hey, yesterday, we ended off talking about the cell cycle, how it can be regulated. In other words, depending on the cell, cells are going to have different times they want to go through cell division. Some cells undergo cell division rapidly, like skin cells. Other cells undergo cell divisions at a much slower pace, like liver cells, and everything in between. Some cells don't even undergo cell division at all, like muscle cells and nerve cells. It just depends on the cell. And so yesterday we said, okay, there's basically this major regulator. And then they're in the form of molecules. We talked about them being cyclin and CDKs, cyclin-dependent kinases. Before I get to those details, remember the big picture. This kind of represents a cell cycle. And what you do need to know is that a cell has certain of these checkpoints, represented by the red blocks. And basically those are like, it almost looks like a knob on a washing machine, and they compare it to that. Being as you got to kind of have everything in check at the end of one cycle, and then before you can go on to a new cycle kind of deal. And the same thing kind of works here, but in the cell it obviously works a little different than your washing machine. That's where this picture is oh so important right here. Understand this picture, make sure it's very clear. You have a couple different molecules here at play, something called cyclin, okay, and then something called CDKs. These are enzymes, and these are proteins. These cyclins are going to start building up in the S phase. Once they do, they are going to start combining with the CDKs, these kinases, these enzymes, because those kinases are just kind of always going to be there. And then they're going to form this unit called an MPF, a mitosis performing factor or something like that. Okay, there's a couple names for it, maturation, promoting factor, whatever. Anyway, that's basically this complex that is going to be responsible for, well, phosphorylating a bunch of molecules along the way to make mitosis kind of progress. That's kind of unfortunately, that's the easy way to say it. The very last thing we were talking about at the end of the class is the fact that towards the end of mitosis, during anaphase, that cyclin will start breaking down. It should start breaking down. And then Jonathan actually asked the question about, well, what happens if it doesn't break down? And that's where it does get complicated. But yes, there's obviously problems that can happen. I actually went back last night and I researched, because I hate not knowing a question. Oh, where did it go? I was researching that same question. And by the way, when it comes to questions that like some of you guys like to ask, you know, your answers aren't going to be found in textbooks. They're not detailed enough. But that's where you, as a budding young scientist, can always go to these scientific journals to get answers. Now, the only problem with these scientific journals are that they're very technical. They are very, very, very technical. This one is actually kind of entitled The Regulation of Cyclin D1 Degradation, Roles in Cancer Development and the Potential for Therapeutic Intervention. And if you kind of continue reading, it gets very, very, very detailed. However, here's kind of what I generally think is interesting. Yeah, there's a connection. There's a connection between the fact that if that cyclin doesn't degrade, that cancer is definitely a possibility. Yeah.
What is it then? There's like it's like psoriasis or something where like your skin cells. Would that be like a cyclin thing?
I don't know if
It's
that's
like-
related to the cell. I don't know if psoriasis is a form of cancer.
It's not, but it's like an overact of like you have too many cells in one spot.
Okay, well, yeah, it might very well be related. I actually don't know. That's a good question. That's a really good question. Huh. And that's where we're headed today. Before we do that, anyway, I'm not going to have you read this, but it's funny how, you know, just some of the questions you ask, the best way to kind of answer some of them are to go to the current research that's out there. This is kind of different science groups or different lab groups, different scientists spend their whole life history, or their whole life just studying the cyclins that are, or not even the cyclins, a certain specific cyclin responsible for a very small, you know, checkpoint in cell cycle. I mean, it's actually interesting how detailed you get when you start getting into the science field and you get into a certain lab, and they really, really start getting very technical about very small and technical about what they study. Okay, well, going back to this, understand this, and I think we have a good grasp of that. Okay. Not only, here I'm going to go back to this for a second before I go to that picture. Not only is the cell cycle regulated internally, like you see in this picture. In other words, all this stuff is happening on the inside of the cell. We're inside the cell. There's a buildup of cyclins. There's CDKs. We're inside of the cell. There's actually also some regulators of the cell cycle outside of the cell. Before I go to the pictures, actually, let me kind of talk a little bit. I'll come back to the pictures. I changed my mind. Okay. In other words, a cell can have all the cyclins in checks, all the CDKs in check. Everything seems to go for this cell to progress and move on. However, outside of the cell, a couple of things also matter. I'm going to kind of just say other factors. And the one thing, I'm not going to go in very much detail about this, they're just called growth factors. These are proteins.
This is other reasons why they can't go forward.
This is yeah, basically here's the deal. A cell could have all those cyclins and CDKs in check and look good. However, if certain growth factors are not available to that cell from the outside, the cell won't go. It won't divide. So these are specialized proteins, and I'm just going to keep it real basic, that stimulate the cell divide. In other words, like I said, if you don't have the growth factors there, it won't have I almost consider it like a medium, like a medium as in like they have to be suspended in a certain. They have to have like a certain medium around them in order for cell division to take place. Okay. Also, one more thing. I'm going to actually other factors, and I'm going to put the word external, because the two things I'm going to talk about here briefly, and I'll show pictures in a minute, are things that have to kind of take place out of the cell for a cell to divide too. So these growth factors have to be there. They're just specialized proteins that are going to get bound to a receptor on a cell, all that to set the stage in motion. The other thing is cells, normal cells, inhibition. Normal cells, and here's where I'm going to start my talk into cancer, but normal cells will actually stop dividing if they become too crowded. Crowded cells stop dividing. That is how cells normally behave. and it's called density-dependent inhibition. Inhibition, you're stopping something. Density, you guys know what density is. Density-dependent. In other words, cells will stop growing if the density, they become too crowded. That's how a normal cell is supposed to respond. Now here's what we're going to see with cancer. Cancer cells don't care. Cancer cells are kind of totally, it's almost like, think of cancer cells as like they take on a whole new set of rules. There are no rules. In other words, just because a cell starts bumping up with another cell, cancer cells will continue to divide even though they're crowding out. They don't sense that. Growth factors, if you're a cancer cell, maybe your immediate thought is, okay, well, let's just take away some of these growth factors around a cancer cell and bam, no more cancer, we'll stop growing. Cancer cells are special in the fact that they think that cancer cells can actually make their own growth factors and keep dividing. Cancer cells are stinky little suckers. Okay, go ahead. Do you have a question?
That was a bug.
Okay. Oh, okay. All right. Let's move on. Remember those. Let's move on to cancer real quick. I don't know if you guys really actually realized technically what the definition of cancer is. It might not be what you think. When you have cancer, it's not like you have a bacteria. It's nothing like that. It's basically when a cell or a group of cells just cannot learn to stop dividing. In other words, all those regulators that we were talking about that kind of, tell a cell when to progress to a new phase of its cell cycle, totally blown out of the way. They don't understand them. Uncontrolled growth of cells. So these are cells that literally just undergo a cell cycle way too quickly, and it's an uncontrolled kind of cycle that they have.
How does it spread to like other parts of the body?
That's a good question. We'll talk about that in just a second. Okay, that's a great question. Were you going to ask the same question?
I think they masticize or something.
Yeah, what actually ends up happening, it's so simple actually what cancer is, but I'll explain that in just a minute. Growth factors are irrelevant. Okay, if they're missing, cancer cells can make their own. They're stinkers. Okay, we mentioned it real briefly yesterday, but not enough. I'm going to put a little star here. Even though we haven't talked about genes and DNA in much detail yet, the underlying basic cause of cancer is there's a bad gene. One of your genes in your DNA gets damaged. But let's remember that if one of the genes in let's say this is a piece of DNA, and again, what a gene is just kind of a small segment of your DNA. So this would be like a gene. By the way, a real famous cancer gene is something called P53. It's just they know that this is a gene that is there to promote cancer. In other words, if you have a lot of genes running all down the side of, or all that making up your DNA. If your gene gets damaged, it is not going to produce the correct protein that it needs to make. And we will go into more detail about that process coming up. So, well, this is kind of what happens. Think about it in cancer. What they believe is that it all goes back to the DNA. You have a faulty gene producing a faulty protein. That protein cannot go on and do its job. Maybe that protein was a cyclin. Maybe that protein was another protein that handles some other kind of checkpoint in the cell cycle regulation. There's actually multiple genes. P53 seems to be a famous one. For the details, I don't know exactly, but it is a very famous cancer gene. When it's faulty, when it's damaged, it's not going to perform its job. If it's not able to perform its job, it's going to, and in this case with cancer, the cell's not able to regulate its cell cycle. So all the genes that are involved in cancer have to do with the regulation of the cell cycle. Yeah.
I was curious, do you know when cancer was first really discovered?
Oh, gosh.
I'm just curious, did it form over time and get worse like the staph infections are doing? Did it become a superbug? Or I didn't know if people in later history had cancer.
Like, are you saying, do you think the cancer rates were lower back in
Yeah.
history and higher now?
Yeah.
Well, that's a whole other discussion we can have. When you start, if you ask my opinion and what I think is coming out in research, I think cancer rates are on the rise now. My personal opinion, but although this is not scientifically based, but just from articles I've read and things that I've kind of taken in, our society now is consuming a lot more processed foods and taking in a lot more environmental, like, you know, we're smoggier, we're eating foods that are prepared and have lots of chemicals in them to keep them preserved. Those are my reasons that I actually think cancer rates are rising, because those have been proven to be, in a lot of articles, causes of cancer, causes that make the gene get broken.
How does it, like, processed food make your...
There's a lot of chemicals in processed food. So instead of going out in the old days like they did and literally like kill the chicken fresh, eat it fresh, no preservatives, nowadays a lot of the food in the supermarkets filled with preservatives to make it last longer.
So naturally, body's not used to it.
Yeah.
I blame pesticides and microwaves.
There's a lot. That's kind of where research is headed. So yes, to answer your question, I absolutely think cancer rates are higher than they used to for kind of those reasons. But even like pollution, smog, you know, we're putting chemicals in everything, water, food. We're even doing things, you know, to our livestock that they never used to do in the past, but for good reasons. But you know what I mean? Right. It's all part of the game. That's my personal opinion, but I hesitate to say that because, you know, it's just from kind of reading stuff. Yeah.
So, like, is that why, like, if you have, like, cancer in your family, is that why that gene
gets passed on?
That bad gene is...
That bad gene, if it gets passed on, you are more susceptible to getting it yourself. Yeah. That's why you see cancer kind of say that it's kind of hereditary in the family. But there's multiple reasons to get cancer. Sometimes it's passed down, but like we said, sometimes you can get it through the environment. It's kind of the nature and nurture thing. So that's why a lot of times cancer happens in older people. Older people have had many years living on Earth, accumulating lots of these little mutations in their DNA. Some mutations don't do anything, but it's usually when you have this accumulation of mutations in multiple locations, that's where you kind of start running the risk of things like cancer and anything else. Yeah.
Oh, I didn't know if there was anything.
No, that's fine.
I don't know how accepted the term cancer cluster is, and I'm not sure, but I think Paulding County is probably got to be on its way to becoming a cancer cluster. And things we have here, pesticides and microwaves. On that now, we can move on. I just wanted to know.
I know, it's funny though, because I have noticed since I moved out here, versus living in the city, granted city water is not wonderful. They have to put so many chemicals in it. But I've noticed the farming that goes on around here. I've never lived in an area where I've gotten so many reports from the water department saying that we've tested your water and there's an unusually high of this and this, and it's probably due to farming, blah, blah, blah. And that's something I haven't had to use to. Granted, you know, in the city you have your own issues. You basically are breathing yellow air, but that's a whole other issue. But it is funny. It's almost unavoidable in a lot of these things. But yeah, that's why I think cancer rates are rising. Definitely. What were you going to say? I absolutely agree.
Or Texas.
Yeah, I agree. I agree. All right, so cancer, may not be what you think it is. Hey, now let's kind of talk about what Arlen's going to talk about. There are by the way, what a tumor is, let's back up. What a tumor is, when you hear, it's literally just a mass of cells. They're not diseased, they're not, they don't have a bacteria or virus in them. They're literally just a clump of cells that have accumulated because mitosis has just gone crazy there. Yeah, a cyst is the same word. Cyst is the same word.
I thought cysts were hollow. No, never mind.
I don't think so, but it sounds like it, but no. There are benign tumors, and then there are malignant tumors. Do you guys know the difference?
One's cancerous.
Benign is cancerous.
Actually, correct. Sort of. Benign. If a tumor is considered a benign, it remains at the site where it started from. Okay. If a tumor is considered malignant, it's spread. it's the same cancer, it's the same group of cells, but the fact that it's now spreading around means that it's malignant. That's when they actually technically call it cancer. I mean, I've heard, well, it depends on who you talk to. Some people will technically only call it cancer if it's malignant, if it's a malignant type tumor, if it's spread. Okay, by the way, a couple other words you might have heard with regard, I can never spell this, metastasize, like if something is metastasizes, it's whatever, but that just refers to the process where you have the spread of cancer cells. So if you say that the cancer is metastasized, it's spread. Okay, that's when it becomes a problem. You know, you could have cancer, and if it remains at the site, you could go in with surgery, bam, done, removed, gone. Okay? It's when it starts spreading that it becomes a little harder to contain, a little harder to treat. Go ahead.
Do the tumors like shut down your body? Or?
Yeah, what ends up kind of getting to you is literally these masses will travel through your bloodstream and literally clog major arteries, veins, other places where things need to kind of go in and out. So you usually have an organ failure due to cancer because, you know, basically if that tumor is there, it's blocking the function of that organ. So that's usually what happens. That's why it's funny. It's funny when you hear, not funny is the wrong word. It always baffles people. Let's put it that way. That they say they started off with breast cancer, but they ended up dying of a different form of cancer, and that's literally because that same tumor that originated in the breast got traveled maybe in the bloodstream, maybe in the lymphatic system, somewhere else, and it ended up in another location, basically shut down the function of that organ.
How can it spread?
The bloodstream. The cells literally seep into the bloodstream and travel like a highway system.
Doesn't it have to be more of just like a collection of cells? That, like, aren't they like disease? Like
they're not. They're literally. It's just a collection of cells. Believe it or not. It's not. Granted, they're not properly functioning cells.
Because doesn't like they say like everyone has cancer in them. Like I read somewhere like it was like this article like everyone has cancer, but some people just it doesn't. Like everyone obviously it's just a collection of cells. Like we all have that somewhere. Like not.
And I'd have to see the article to see how it's worded, but I can understand maybe what they're saying.
It's like we all have it, just it doesn't.
Yeah,
some people it doesn't affect. Like, it doesn't collect in the way that it does in a cancer person.
We're also going to learn. Well, and that's going to get way too detailed, but I
want to be able to find that again because it was weird how it worked.
That's interesting. I don't know. Yeah.
Is it true that moles on your body hold cancer?
Depends on them. What moles are just kind of like skin cells that have just kind of, I don't want to say the word mutated. When I say the word mutated, don't think that all mutations are bad. So, but they're just kind of have become differently growing cells. They have since they're odd to begin with, I think those cells are more susceptible to becoming cancerous, but moles in themselves are not.
I was raised by my great-grandma, who's nearly 90 now, and she's always, don't touch them, they're cancer.
They are not when they're on you, but they have a lot of times they say they have the ability to become cancerous, but moles in themselves are not. My daughter has a little mole on her knee, and her doctor said it's fine for now, but she says once she hits about puberty age, she's 8 right now, that they might have to consider removing it. So I don't know if the hormones play a factor in maybe stimulate, that is, I don't know. But, I don't know. Yeah.
Yeah, my mom would, she used to be a cancer nurse, she would work in that area, and now, she's always, after our moles get so big, we go and get them removed because
Chances are, as you get older, you will have moles removed. I mean, it's pretty much a given, I think, for everybody. That's why, it's a given. It's a given. Yeah, and they can tell the difference. Most of them are not. It's when they start becoming irregularly shaped and raised and red and weird and odd. They actually look for the ones that are round. The round ones are okay. That's what I've heard. All right, guys, a couple other things. Briefly, this is more for radiation therapy. Radiation therapy versus chemotherapy. I don't know if you guys realize this. I figure it's good to know. radiation therapy, when you have a mass of cells and they're growing out of control, they're growing uncontrollably, a lot of times you can go in with radiation and pretty much kill those cells in a very localized spot. That's why radiation therapy is basically using radiation that's basically sent to a very localized place. I assume this is in the form of lasers, some kind of, I imagine it's a light radiation, like light ray, I don't know. That's different from chemotherapy. Chemotherapy, the word chemo kind of means chemicals. This is where you're actually injecting substances to the spot of the cancer to kill the cells. Now, both of these have side effects, as you guys have probably seen, hair loss, nausea, all these things, because I think both of them pretty much do. Both of them have hair loss, both of them have nausea. A lot of them make you sick, like ill. And the reason for that is that obviously if you're injecting yourself with radiation or chemo or chemicals, it's not only taking out the bad cells, but it's taking out some of the good cells. So that's kind of what happens with the side effects.
Do some people do both of them?
I don't know. I don't know. I've always heard if you're only doing one at a time, but I've never really known anybody to have cancer close to me. So I'll be honest, I've been lucky, knock on wood, so far with that.
When you get leukemia, which is a form of cancer that affects white blood cells,
it's in the blood.
Do those just spread excessively?
Yeah, that's why leukemia is a lot harder to treat because it's in your bloodstream and it's moving constantly. So that's why it's very much a form of cancer that's very difficult to treat. Some cancers are easier to treat than other ones, and leukemia is a tough one because it's spreading.
Start like in your bone marrow?
Yeah, because that's where your blood is made in your bone marrow. Very good, and that's basically where it begins. It's made and then spreads.
Yeah, my grandpa was like my grandpa that died from. He was told he had it, and then like 3 years, 4 months later, he was dead.
Oh wow.
I mean, it spreads.
That was a great story.
Chemotherapy's been around for a long time, right?
It really has. I mean, you've actually heard of both of these for quite a while, but
I'm just surprised that chemotherapy is still the best option in the medical community for treating cancer, and there's nothing better.
I don't think
I'm assuming what they're doing at this point now that they've. Oh, this is a whole other issue. Now that just recently, within the last 10 years, we've there was a big project. I did want to bring this up to you guys anyway. It's a good one called the Human Genome Project. If we can get through with this, I may actually show you the movie on Tuesday if we can get through some of this. There was one of these. This was probably within the last 10 years the best breakthrough in. It's the movie I had. Oh yeah, basically they mapped the human genome. Is what the word is. Genome. A genome is just a fancy word for all collectively everything that's contained in your genes. They basically mapped all the A, T, C's, and G's that are found in human chromosomes. Okay, so they basically there's like 3 billion of them. So they basically found out all 3 billion A, T, C's, and G's. That's kind of where they got that map from. That's exactly right. So that was actually a big deal because now that they have this, now I think they're going to be able to do different treatments. They're going to go right to the source, right to the gene involved, and try to fix it at the gene source rather than trying to fix a cancer even while it's there. It's almost like fixing it in advance. So that
last year in this is weird chemistry and biology, Jared Ackman's mom came in and talked about that.
Okay.
Well, I don't remember what she did. She does something. She usually works in a lab. Yeah, some sort of.
Are they gonna try and like take that gene out? Is that what their goal is?
A lot of what you can do with the gene if it's a gene that they know is broken, a lot of research is out there trying to fix those genes. Or if it's a gene that's making a protein that's messing things up, they want to go in and be able to shut down that gene's. When I was in a lab, I don't know if you guys realize this, when I worked with tomatoes, it's kind of a joke, but when I worked at the USDA and I worked with tomatoes, that's exactly what we did. We were going into tomato DNA and we knew where the genes were, and there were multiple of them. The genes were that controlled the ripening of a tomato to make it soft. And we were going in there and cut and pasting those genes out, turning off their functions, plugging them back in, and trying to make it so that tomatoes don't ripen as fast. That way you can take them to market. They can sit there forever rather than only sitting there for about a week before they get all soft and mushy. So that's exactly what that's what genetic engineering is. Basically going into a gene, fixing it, or cutting it out, or turning off its function to hopefully, that's exactly what genetic engineering is.
It's like cleaving, right?
Yeah, it's literally like cut and paste. Now, the only problem with that, we're going to learn when we talk about DNA, is, number one, I told you the genes that control the ripening of fruit, there's many of them, okay? And then also, those genes not only control the ripening of fruit, but they do other things in the tomato as well, so you're screwing up those functions.
So there's side effects.
So there's lots of side effects. It's like, okay, there's 7 genes that control the ripening of tomato fruit. I'm going to go in and turn all those 7 genes off, but not only have I fixed that problem, but I've caused 10 more because those genes also did another job. Yeah. So that's why it's complicated. It sounds so easy to fix, but theoretically...
Did it work then?
No, I made some really crazy looking plants. Oh, you did. You actually, when you start going in and mutating or changing, genetically engineering the DNA, I made some of these plants that we ended up making are, they had tomatoes on them, but they grew like ferns. So the rest of the leaves looked fern-like.
So
Then you would, and then other ones, tomatoes, you would cut open them up, and the insides would look like sand instead of like, it was just, in other words, you're messing up with other proteins being made when you go into mess with genes. It's really neat. It's neat. So did it work? Science is such a slow progress. I was only there for 3 years, and I hardly made a dent in anything.
It's like you don't even work on.
No, I was actually not the peon. I was the peon in the group of 7 of us or so at the USDA. Yeah, I was definitely the peon.
Did you create any tomatoes that like could crawl or walk?
No, it wasn't like Attack of the Killer Tomatoes.
How do you exactly like find that in the human though? Find that gene that.
We will discuss that when we head into that chapter. Okay, we will talk about it. But that's what research is going on right now in that field. Is now that we figured out the parts list of the human genome, we know where every gene is, like on that map, on that picture. Now the question becomes, okay, we have this parts list. Now we need to kind of find ways to manipulate these genes if needed. However, guys, that raises a whole bunch of ethical issues too, as you could probably imagine.
So then if you change like in the embryo, or would it be like?
You could. You could. And that's why it gets into, are we trying to create a superhuman, or are we going to evolve our species into something that is not human anymore? Yeah, I mean, it's very...
better.
No, but there's a whole lot of questions that can be raised from
It's
that.
kind of scary though.
Very.
With you guys just made the plants look different. What would it do to a human?
Exactly.
Have
I'm
you tried
sure
that?
I'm sure there are labs doing it, but I think it's illegal to...
There's underground tunnels in America that lead to places that.
You're just kidding.
No, I'm serious.
Okay.
They try to breed humans with animals and like find
Bestiality.
genetics.
And just so everyone knows, Caleb also thinks that we paid Japan to attack Pearl Harbor.
This is on video. We don't need to discuss this right now.
We don't want to embarrass anyone.
We don't need to have
I didn't hear.
an
That's all
argument.
right. Hey, are there any questions? That was chapter 12. I'm actually done with chapter 12. It was basically the big points about it are cell division. We talked a lot about mitosis, but again, remember, mitosis is such a small part of the life cycle or the cell cycle. It's just division of the nuclear material, just the genetic material. Then at the end of the end, we kind of talked about how that cell cycle can be regulated, kind of controlled, like an on-off switch in a lot of ways. And we definitely talked about those cyclins and CDKs. And then they talked about basically kind of what can happen if the cell cycle is not regulated. And that's kind of why they brought up cancer. And because that's basically what it is. It's uncontrolled growth of cells due to like a lack of control of the cell cycle. Yeah, those checkpoints just kind of get thrown out the window. So understand the big picture, guys, again. Are there any questions you want to, anything else that didn't make sense?
This is like a random question. Like, so then
No.
like, there's 2 strands of DNA, like when it's replicated or whatever.
Yes.
Can it make protein off both of them, or does it just work off that? Like, like pump out double the protein, or?
Oh, I never really thought about that.
I don't think they're making proteins when they replicate. Yeah, I guess it would be doing. Yeah, you're right.
I guess theoretically they could, because remember that replication cycle happens in the S phase. So yeah, they're still going to be producing proteins all throughout. There are lots of on-off switches for DNA as well, though. That's what we're going to talk about. So it must have something to do with that. Huh. Good.
Chapter 13.
Yeah, let's go to chapter 13. Lucky 13. All right. This is all about sexual life cycles. Okay. And I'm going to put in parentheses, and meiosis. But again, it's just going to be kind of a part of the picture. Meiosis is, I just right here, it's a special kind of cell division. It's still cell division. That's why I wanted to include it along with the last chapter. It is a cell division, but it's very, very, very different. Okay, and by the way, we are headed into the world of DNA eventually. I'm going to kind of draw, you don't need to draw this again, but I have it in my notes to just remind you that DNA is made up of a bunch of genes, and those genes go on to code and make for proteins, okay? I mean, I know I just wrote it. Basically what protein, or what a trait is, a certain trait about someone, like it's blonde hair, blue eyes, susceptibility to cancer, whatever, is basically an accumulation of all these proteins. So these proteins are going on to do a job or to express a trait, very much so. That's kind of how it all works. It all goes back to your DNA. Some key terms I think you guys are already aware of, but let's go over them again. Remember, gametes refer to sex cells. I might have already wrote this down. Remember, somatic cells were the body cells, but this chapter is really going to focus on sex cells in a great deal. I do want to take, it makes for a great test question on AP or otherwise. Basically, key differences between asexual reproduction and sexual reproduction. We touched on them a little bit last chapter, but I want to kind of, for the record, make it official. Last chapter, remember, we talked a little bit about asexual reproduction because there are actually some organisms that can reproduce. They use mitosis as a way, and cytokinesis, as a way to actually reproduce and make more of themselves. So, in other words, some organisms reproduce asexually by mitosis and cytokinesis. Cytokinesis. Because remember, after mitosis, you technically don't have two new cells yet. You need them to split by mitosis. Some protists actually reproduce asexually this way. Budding. We mentioned it briefly. That's another form of asexual reproduction. What was the name of the method that bacteria use a lot of times? Binary fission. That's definitely another form of asexual reproduction. And we went into a little detail last chapter. So asexual reproduction we've already talked about in some detail, but it is a form of reproduction in certain organisms, as well as some of these other things. All of these methods pretty much result in making a clone. A clone is just a genetically identical cell. A genetically identical cell, clone. Well, that's easy to remember if you're a Star Wars fan because they have the Clone Wars movies, which my husband is totally into. So, you know, clones are genetically identical cells in this case, but
They're
only
like genetically identical, right?
Yeah,
they can look different.
You wouldn't look different if you're genetic to the same, would you?
Like
You can, because the environmental, the way that that gene is expressed can be different from one person to another person. So technically, yes, but for the most part, a clone is something that's genetically identical. But no, you're right. There are actually environmental factors that can go on.
Are we cloning a person?
Not that we're all aware of.
It's happening.
And like I said, I think if it's happening overseas.
Conspiracy
It's not happening in the
theories.
United States. They have less regulation when it comes to that stuff overseas. It very well might be. It wouldn't surprise me.
Underground in those tunnels, they exist. They exist, John.
Some of the aliens?
They exist too.
No, they don't.
Yes, they do.
Here?
Aliens, they exist.
They don't.
Men in Black.
They
Is that
don't
real
exist.
too?
No.
Okay.
Hey, when you start messing up the genetic structure, when you get a unique combination of genes, and you get those unique combination of genes from two parents, that's basically what makes that different from sexual reproduction. Okay, this is probably nothing earth-shattering. You guys probably have known all this. I'm not trying to trick you with anything. What is important, though, is both of them have advantages and disadvantages. I'll go through those definitely. Asexual reproduction has definite advantage in the fact that it's faster. Okay? In other words, when a bacteria decides it's time to divide and make more, they don't need to seek out a mate. They don't need to exchange genetic material. It's not an energy-consuming process. They just split. Faster, less energy. Yeah, they don't have to. Yeah, they don't have to woo anybody with a dance or anything like that.
It's also more efficient in the genes, right? Isn't it? Like, there's not as many hiccups.
Oh, I guess you could think of it that way. But now, I think the mutation rate is pretty much the same regardless.
I always thought that if something asexually reproduced, it would be almost exactly the same as the parent.
No, the mutation rate in organisms that reproduce asexually is pretty much the same as the mutation rate of those that is sexual reproduction too.
Is budding does it just grow off the side then? Yeah,
it's literally like a mass of cells just kind of grows by mitosis kind of off the side and it kind of breaks off and forms. Yeah, very yummy. Disadvantage of asexual reproduction. I don't want to say that. Let me actually put another advantage. Asexual reproduction has a definite advantage for those organisms that are living in a stable and non-changing environment, which really technically never happens. But if you're in an environment that doesn't change, asexual reproduction is awesome. Sexual reproduction, however, has an advantage in a changing environment. And I'll be honest, guys, the environment is constantly changing realistically. You could trace back to Earth history and just see how, even weather, environmental, everything conditions alone have changed. But it's more than just that. Think of different environments are changing on all the time, even in a shorter period of time.
For a fetus inside of the womb, is the environment really changing that much?
Not necessarily probably in there. I'm talking very much in an external environment when it comes to temperature and weather and chemicals and maybe a fire that comes along or maybe a new predator that comes along or something like that. Okay. Genetic diversity is very important in this sense because let's kind of give an example. A bacteria that is going to reproduce asexually. Let's say they're going along, living, doing their binary fission, making lots of bacteria, multiplying like crazy. Life is good. However, maybe a certain strain of a virus or something comes along. Okay, well, if that virus is going to take out one of those bacteria, it's going to take them all out. They're all clones. They're all genetically identical. Okay, however...
How do they adapt to, stuff, though?
The virus or the
bacteria?
In that situation, if they're all genetically identical, they have a harder time doing that.
Yeah, aren't they more adaptable? Like in hospitals and stuff?
The reason that they're a problem in hospitals is just because their reproductive rate is so high. Okay, bacteria. Now, some bacteria, though, that's a whole complicated answer. Some bacteria can actually change genetically, and the ones I'm referring to are the ones that cannot. Okay, so I'm referring to bacteria that never change genetically. Sexual reproduction. Okay, so let's take the same population of something. Let's go away from the bacteria for a minute. Let's take this population of, I don't know, insects maybe of butterflies. They reproduce sexually, so what's going to happen if their environment around them changes? If there's a big population of butterflies in an environment, their population changes and this virus comes in. Well, they're not all the same, so that virus, chances are that virus will not take out all the butterflies. They'll maybe just take out part of the population. Now, yes, part of the population of butterflies will die, but the species will still continue on. And that is kind of where sexual reproduction has an advantage for a species, maybe not necessarily an individual, but a species. So species are much better off with sexual reproduction in a changing environment.